Hello, and welcome to the Science, AAAS, and Science Signaling webinar. My name is Annalisa Van Hook, and I'm the web editor at Science Signaling. In today's webinar, we'll be discussing the role of the tumor microenvironment in promoting cancer progression and impeding treatment. This webinar is the second in a series focusing on the signaling pathways that allow cancer development and progression, the emerging research in identifying and targeting these pathways, and innovations in developing cancer treatment options. Tumors are complex tissues in which the cancer cells evolve and communicate with their surrounding microenvironment. These tumor-stroma interactions are important determinants of tumor survival, growth, and dissemination. This webinar will focus on how the tumor microenvironment promotes oncogenic progression and protects the tumor from therapeutic intervention. By better understanding the tumor microenvironment, researchers can develop strategies and treatment options that neutralize its oncogenic influence and more effectively attack the tumor itself. It's my great pleasure to introduce our guest speakers for this webinar. They're Dr. Rakesh Jain from the Harvard Medical School and Massachusetts General Hospital in Boston, and Dr. Padmini Sharma of the MD Anderson Cancer Center in Houston. A very warm welcome to you both, and thank you for being with us. Before we start today's webinar, I have some information that our audience might find helpful. Up to the right of your screen, you'll see our speakers. Click on the View Bio link to see more details about each of them. Under the slide window is the Resources tab, where you can find additional information about technologies related to today's discussion, or the link to download a PDF of the slides. Our speakers will give short presentations, after which we'll have a Q&A session in which they'll address some of the questions submitted by you, our live online viewers. So if you're joining us live, start thinking about some questions and submit them at any time by clicking the Ask a Question button below the slide window, typing your question into the message box, and then clicking OK. Please remember to keep your questions as short and clear as possible, as that will give them the best chance of being put to our panelists. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant widgets at the top of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology and the Koch Institute for their sponsorship of today's webinar. Now I'd like to introduce our first speaker for this webinar, Dr. Rakesh Jain. Dr. Jain is the Andrew Wirt Cook Professor of Tumor Biology at Harvard Medical School and Director of the Edwin L. Steele Lab of Tumor Biology at Massachusetts General Hospital. Dr. Jain is a member of all 3 U.S. national academies: the National Academy of Sciences, the National Academy of Engineering, and the Institute of Medicine. His research has focused on the idea of normalizing the microenvironment as a new strategy for treating disease that's characterized by abnormal blood vessel growth. Welcome, Dr. Jain.
Thank you, Dr. Van Hook. I would be just pleased to discuss in my next 20 minutes the complex tumor microenvironment of the tumor and show evidence from both animal models as well as patients that every component of the tumor microenvironment is abnormal, and this abnormality fuels tumor progression and treatment assistance. And I'll present 2 strategies on how to normalize the blood vessels and matrix of a tumor to improve standard of care therapies as well as immune therapies. As shown in the next slide are my conflicts of interest, which is slide lists my potential conflicts of interest around the Scientific Advisory Board, equity, and I will discuss off-label use of a number of drugs which are listed in the slide. So the next slide shows how we visualize microenvironment of a tumor, which is pictorially shown here, where a tumor, we have been studying tumor as a complex organ, and this organ is composed of not only cancer cells, which are shown in gray color, but a variety of host cells that includes fibroblasts, myofibroblasts, resonant and transiting immune cells, and you'll hear about that a great deal from Professor Sharma after my presentation. And these cells are nourished by blood vessels and drained by lymphatics, and they all are embedded in extracellular matrix. What our work of nearly 40 years has shown that each of these components of the tumor microenvironment are abnormal and how they fuel treatment assistance. And what I'm going to show today is how we can reverse some of that by de-engineering the tumor microenvironment. So let me first show you, give you the evidence of this abnormality in a live animal model using something known as intraviral microscopy, where we have developed a number of custom-made microscopes and imaging technologies that allow us to visualize molecular, cellular, and functional events in tumors going in various places in animals. And we can see through these glass windows placed on these animals various molecular parameters, cellular parameters, anatomical parameters, and functional parameters during the growth of a tumor or its response to a therapy or when it becomes resistant to various treatments. And as shown in the next slide is when we look through a window in the brain of a mouse, we can visualize the blood vessels in the normal brain, and in the upper left-hand quadrant, we can see the blood vessels of glioblastoma, one of the most deadly human tumor. And just a casual observation suggests that these blood vessels are different from the normal blood vessels in the brain of this mouse, and we're going to learn a little more in a few minutes. But what I want to point out here is this is true not only for transplanted tumor, which is shown here, but is also true for spontaneously arising tumors in genetically engineered mouse models. So, for example, this is the colon of an APC mutant mouse, and you can see the blood vessels in this are abnormal. And this is an 11-week-old mouse. Now, a couple of weeks later, the blood vessels become even more abnormal. And then if you wait 3 more weeks, a 16-week-old mouse, you see the blood vessels are even more abnormal. And you can see some regions one cannot see even blood vessels at all in this particular case. So the question is this an artifact of transplanted tumors or spontaneously arising tumors? So to answer this question, we have also made vascular casts of human tumors. And this is a cast of a 1 kilogram, or half a kilogram, or that is about 1 pound colon cancer from a patient. And this is a polymer cast, so you can see blood vessel abnormality. And the lower micrograph shows that higher magnification, you can see the same kind of abnormality in these vessels as we saw in the tumors, which are in animals. Now, if you look at that higher magnification at these blood vessels, using something called the multiphoton microscopy, what we see is the following, that on the right-hand side of this image, the blood vessels seem pretty well perfused, but on the left-hand side, it looks like there are no blood vessels. But if you were to take this tumor and make a cross-section here and use immunohistochemistry, we'll find the blood vessel density on the left is approximately the same as the blood vessel density on the right. So what is going on? To find out what is going on, we need to look at this whole thing in real time, and as shown in the next slide, we see blood flow in this particular tumor as a function of time, and you can see the flow is quite brisk in the blood vessels on the right-hand side, but it has essentially stopped on the blood vessels on the left-hand side. So what this suggests to us that these regions of the tumors which are on the left region of the tumor, they're going to have a poor oxygen delivery, poor nutrient delivery, and it has going to have its own consequences, which I'll discuss in about a minute. But the vessels which are present on the right-hand side that are perfused, they also have an abnormal function and structure, and that is they're leaky in focal spots. And as a result, they transmit the fluid pressure from inside the blood vessel to the outside, and they raise interstitial fluid pressure in tumors. And we have made these measurements not only in animal models, but in about 150 cancer patients. And as shown in the next slide, we see that these blood vessels, the fluid pressure is elevated in patients who, unlike normal skin or normal breast, where the pressure is around 0 millimeter of mercury, and every primary or metastatic tumor, we have seen the pressure goes up. And this elevated pressure also aids tumor progression. But the bigger challenge, really, which comes from abnormal blood vessels, is hypoxia. And as shown in the next slide, hypoxia and low pH, which is also a result of poor perfusion in tumor, has multiple consequences. The most important consequence of hypoxia leads to inflammation, immunosuppression, and fibrosis. And it upregulates molecules such as PD-1, PD-L1. These are the checkpoint inhibitors you will hear about from Professor Sharma in the next slide, in the next presentation. But in addition to causing inflammation, immunosuppression, fibrosis, hypoxia can lead to resistance to various therapies, standard of care therapies, as well as emerging therapies such as immunotherapy. It can induce cancer stem cell phenotype. It can do EMT programming, cause invasion, and increase metastasis. It can increase resistance to death with various mechanisms. It can change the metabolism of the tumor, and hypoxia can also lead to genomic instability and unfolded protein response. And finally, hypoxia can lead to more abnormal blood vessels. So given all these adverse consequences of hypoxia, low pH, and high interstitial fluid pressure, our laboratory began to examine about 20 years ago why there is hypoxia in tumors and how can we alleviate it. So if you think about it, the reason any tissue has hypoxia is if the blood supply to that organ is poor, or if that organ is consuming too much oxygen. So we decided to focus on blood supply. So to understand why blood supply in tumors is impaired, let's just look at a very simple schematic shown here in the next slide. This is a watering hose, and you're essentially, what you're doing is watering your lawn, and suddenly water stops flowing through this hose, through this tube. So the question is, where would you look for? What are the potential reasons for why this flow would shut down? Well, one of the most simple reason is that somebody poked a hole in this hose. And in our body, the molecule that pokes these holes in blood vessels is known as vascular endothelial growth factor, which was originally discovered as vascular permeability factor by my colleague at Harvard Medical School, Dr. Hal Dvorak, and later cloned by Dr. Napoleon Ferrara as the VEGF. And when we place VEGF on blood vessels, they become leaky. And when a vessel becomes leaky, the flow slows down or shuts down completely, depending upon the leak. So when we realized this, and realized this actually in 1988, I've been looking for a way to plug this leak. Now here's the challenge. VEGF causes leak, but VEGF is also survival factor for endothelial cells. So if you remove too much VEGF from tumors, then we would actually starve this tumor. We would actually destroy this blood vessel. So we need to find the sweet spot. And this is what we refer to. We refer to this concept as vascular normalization, as shown in the next slide. Our idea was that in a normal vessels, the blood vessel, in a normal tissue, the vessels are well organized, so there's adequate amount of oxygen and no hypoxia. In tumors, due to abnormal vasculature, there's hypoxia. Now, if you were to give anti-VEGF therapy to these patients or animals, there are three possibilities. One is that the blood vessels are totally refractory, shown in the middle, and therefore no flow will change. So if you look at the flow meter, there's no change. Other possibility is that we give just the right amount of anti-VEGF antibody or agent so that the leakage is plugged a little bit, and then the blood flow will go up. But if you were to give very high doses of anti-VEGF therapy, we would really actually starve these blood vessels. We'll destroy them. And that is shown in the bottom slide, where you see a decrease in perfusion. So the question is, does this happen in animal models? And more importantly, does it happen in cancer patients? To make the long story short, for nearly 10, 15 years, we have shown this in a variety of animal models that indeed this is the case. And we have also carried out about 25 clinical trials in the last decade to try to test this hypothesis. And let me show you the result of one such trial where we actually see this. And what is shown here is the result are the perfusion in tumors, in newly diagnosed brain tumors, glioblastoma. And this is a small trial which had 40 patients. When these patients receive an oral anti-VEGF agent known as cedarenib, in about 20 patients, the perfusion actually goes up. In 10, it's stationary, and in 10, it goes down. Now, the prevailing hypothesis has been that if you starve the tumor, we would benefit the patient. But what I just described to you, if you starve the tumor, you'll increase hypoxia, and you cause immunosuppression and all the other problems. So according to our hypothesis, the patients where the perfusion goes up should be the one who should have a longer survival. And indeed, this is exactly what we see in these patients. The 20 patients where the blood perfusion went up as a result of anti-angiogenic therapy, which was originally designed to starve the tumors, when the perfusion went up, these patients had a median survival, which was about 9 months longer compared to the patients where the perfusion was stationary or went down. And now we have seen this in a number of other trials. In the interest of time, I will defer to that in the question-answer period. But really, the question we are asking right now is why is it that in these patients, we are not seeing an increase in perfusion? And our hypothesis is that perhaps the doses of anti-VEGF agent we are giving to these patients is too high for the amount of VEGF these tumors have. So if you could lower the level of VEGF, perhaps you would be able to increase perfusion in these patients too. If you lower the dose of anti-VEGF agent, we would be able to do perfusion. So we have tested this concept actually in animal models, where we have taken animals with breast cancer, and we have given them either a control IgG, or we have given them an antibody at 40 milligram, which anti-VEGF receptor 2 antibody 40 milligram on the bottom right-hand side, 20 milligram at the bottom left-hand side, and 10 milligram at the top right-hand side. And what we see here essentially is that the animals which receive the lower amount of anti-VEGF receptor 2 antibody show increased perfusion in their tumor. So our hypothesis is that perhaps if you could tailor the dose to a given patient until the perfusion is increased, we may be able to benefit a larger fraction of patients with a standard or currently approved anti-VEGF agent. So this is the concept I've tried to summarize here, where the dose of anti-VEGF agent is important. If we give very high dose, we may cause excessive pruning of blood vessels and lead to hypoxia, shown here in this magenta color. But if you were to give lower dose, there would be a window of normalization during which chemotherapy, radiation therapy, and immunotherapy would benefit these patients, and I'll come to that point later. But what I want to point out is that a variety of human tumors, such as pancreatic ductal adenocarcinoma, do not respond to anti-VEGF therapy. So what is going on in these tumors? Our hypothesis is that these 25% of the tumors are highly desmoplastic. So to point out how desmoplasia can shut down blood flow, let's just go back to the same pose which I had described to you earlier. We are watering our lawn and suddenly water stops. And you look around, you do not see any leak here. So what would be the potential reason why water would shut down in this tube? Well, one possibility is that somebody stood up or dropped a ball on this tube. And when you do that, one would blood flow or would shut down that blood vessel, or water will shut down this tube. And if you could figure out what this ball is and who's dropping it, we could perhaps remove it. And by doing so, we may be able to increase perfusion. And our work of nearly 20 years has shown that it is the blood vessels in tumors are compressed by not only cancer cells and stromal cells, but more importantly by the matrix they produce, high amount of collagen and hyaluronic acid. And for nearly 20 years, we've been looking for ways to alleviate this stress, to alleviate this force generated by matrix on these blood vessels. Now, I want to point out here that these cells and matrix also send out signaling molecules that can affect the behavior of a tumor. But in addition to biochemical signaling, there's a physical force they apply, which we refer to as solid stress. And we've been looking for ways, translatable ways of alleviating the solid stress. And what we have found is that tumors which have very high amount of collagen have compressed blood vessels. And we've been looking for ways to remove collagen. And we have been looking for ways to visualize this solid stress. And the way to visualize this solid stress, if you take a tumor in this movie and you just cut it like this, which I showed you, and I'll repeat that movie again. You see that cut this? You can see that due to the solid stress, this tumor swells up, it opens up. And the swelling and the shape after swelling can be used to calculate the amount of stress being generated by tumors on their blood vessels. And by using a mathematical modeling and looking at the shape after cutting this tumor like this, we learned that the pressure applied by tumor on its blood vessels is about 10 millimeter mercury, 20 millimeter mercury, 30, 40, 100, 150 millimeter mercury. And we have been trying to reduce this pressure, and what we have discovered in the last 10 years is that the widely prescribed antihypertensive drugs that are used to lower blood pressure, and these drugs known as angiotensin receptor blockers, actually block the signals downstream of angiotensin receptor 1, and they block signaling from TGF-beta, and as a result, they deplete collagen in these solid tumors and hyaluronan. And as shown in the next slide, when we do this, you can see in the highly desmoplastic tumor on the left-hand side, these are the collagen fibers imaged using something known as second harmonic generation microscopy. And when we treat these tumors with losartan, with an off-patent antihypertensive drug, in about 10 days, you can see that most of collagen is gone. And when that happens, the blood perfusion comes back. And as shown in this slide, the green color represents perfused blood vessels in this region of the tumor on the left-hand side, and blue color represents collagen. And notice that in the blue region, there are hardly any perfused blood vessels. But when we treat this tumor with losartan, we find out that a lot of blood vessels now appear in this region, which are perfused. And when this happens, we can improve the drug delivery. We can also improve the response to therapy, and the animals survive longer. But the question is, what about patients? So while one of our colleagues has initiated a clinical trial at Mass General Hospital based on our clinical findings, if you look at some preclinical published data on retrospective studies, what we find is that there are 4 retrospective studies which show that when patients, if they take an angiotensin receptor blocker or an ACE inhibitor to control their blood pressure, while they are taking chemotherapeutic agents, whether these patients are pancreatic ductal adenocarcinoma patients, lung cancer patients, or renal cell carcinoma patients, the survival in these patients improve by several months compared to patients who do not receive this kind of antihypertensive drug, or the patients who receive essentially, say, beta blockers. So currently, this hypothesis is being tested in cancer patients, and more importantly, we are developing drugs so that they do not lower blood pressure of patients, but they actually deplete collagen matrix in these patients. So the last point I wanted to convey is shown right here, that vascular normalization as well matrix normalization can also enhance immunotherapy. And the reason immune therapy, as you'll learn more in the next lecture by Professor Sharma, one of the reasons immune therapy is dysfunctional, does not work in tumors, is because they have an immunosuppressive microenvironment. And hypoxia is a major contributor of this immunosuppression. When blood vessels are dysfunctional, various variety of immune cells do not get into the tumor, or even if they do get into the tumor, they stop functioning when they see hypoxia, low pH. A type of immune cell known as macrophage, they become pro-tumorigenic when there's hypoxia, and the list is very large. A variety of checkpoint inhibitors go up in tumors when there's hypoxia. So our hypothesis is that if you could normalize the blood vessels, or the matrix, or both, depending upon the tumor, we should be able to alleviate hypoxia, normalize pH, and normalize the matrix so the immune cells can penetrate the tumors very nicely. And what that will do is increase the delivery of tumor immune cells. It will also make them more effective, and the macrophages will become from pro-tumor to anti-tumor type. We have tested this hypothesis using vaccine, as well as we're currently testing it using checkpoint blockers, and we are seeing this concept when we normalize vessels, these therapies seem to work better. So let me end my presentation just by summarizing the key points, and that is that abnormal blood vessels and abnormal matrix in a tumor create a hostile metabolic and mechanical tumor microenvironment in addition to signaling, aberrant signaling. And this environment is characterized by hypoxia, low pH, and high interstitial fluid pressure. What I also shown you, and others have shown in the literature, that these abnormalities fuel tumor invasion, immunosuppression, inflammation, metastasis, and they induce treatment resistance. What I've also shown you is 2 strategies to re-engineer the tumor microenvironment. One we refer to as vascular normalization using judicious doses of anti-angiogenic agents. And the second one, which is an emerging approach, matrix normalization, where we have shown you how commonly prescribed antihypertensive drugs can help normalize or re-engineer the vasculature. What I've shown you that this approach can improve therapies, standard of care therapies such as chemotherapy and radiation therapy, as well as emerging therapies such as immune therapy. And the very last point, which I did not have a chance to show, that this normalization concept has implications for about 500 million people around the world who have diseases which are characterized by abnormal blood vessels, such as macular degeneration, which is a leading cause of blindness, schwannomas, which is a leading cause of hearing loss, and we just published a paper last year showing that this approach can also help tuberculosis treatment. Just want to end my presentation by pointing out that we have an extended course on tumor microenvironment, which the 30th, we'll be teaching it for the 30th year this year at Harvard Medical School, and October 5 to 8, and the faculty members of this course are listed here, along with the website where you can go and find out more about the course and register. And finally, some resources. My lecture was based on these 5 review articles, one in Scientific American, one on Cancer Cell, which came out in November, an annual review of the bioengineering, which has a lot of mathematics, and this Cancer Cell review, which came out just last week. And finally, a review article on how normalization can improve, enhance immune therapy. I want to end my presentation by acknowledging my colleagues in the Edwin L. Steele Laboratories, especially 8 PIs who are 4 associate and 4 assistant professors who have been my collaborator for nearly 2 decades. And I've had the honor and a pleasure of working with equal number of colleagues at Harvard and elsewhere, and graduate students and postdocs. So thank you very much for your kind invitation. After Dr. Sharma's presentation, I'll be happy to answer any questions.
Thank you for your presentation, Dr. Jain. Our next speaker today is Padmani Sharma. Dr. Sharma is a professor in the Department of Immunology and the Department of Genitourinary Medical Oncology at MD Anderson Cancer Center. She's a medical oncologist and an immunologist, and in her research, she investigates the mechanisms involved in the immune system's anti-tumor response. The goal of her research is to find ways to enlist the body's own immune response to fight tumors. Dr. Sharma, welcome.
Thank you so much. I'm happy to participate on this webinar today. So also thank you to Dr. Jain for introducing the topic of immunotherapy. It makes my job a little bit easier now. Just so that we can do a little background, it's important that we understand the immune response and the things that we would like the immune system to do. We know how to mobilize the immune response to clear infectious disease. It's your immune system and your immune response that can help get rid of bacterial pathogens or viral infections. And so it's very important then for us to say, if we can do that, if we can get rid of virus and bacteria, why can't we get rid of cancer cells? Because typically we should think of the cancer cells as also as foreign. In order for a cancer cell to form, it's mutated. It has many genes that have changed over time, and so those mutations can be seen as antigens for the immune system to then reject the cancer cell. So in this slide, I'm just showing on the left-hand panel, there's a cancer cell in a dish, and you can see the T cells are smaller components of it, and the T cells can eradicate the cancer cell. So T cells are very powerful in that they can adapt to any antigens that are being expressed by the cancer cell or by the pathogen. They have exquisite specificity for those antigens, and then they can develop memory cells so that once you have a response, those memory cells can then come back very quickly and regenerate the response if the antigen occurs again. So for us to decide then, for us to actually harness or unleash the T-cell response, we have to understand what is it about this T-cell response that allows for tumor rejection. And initially there were studies that were conducted in human tumor tissues to look at what kind of cells were really within the tumor microenvironment. Within the tumor tissues, you can see here in panel A, these are bladder cancer tumors. In panel A, you can see there are very few T cells, or these little brown dots. And in panel B, there are many, many more T cells. These are exactly the same types of tumors in panel A and B, but their difference is the number of T cells that are in there. If you look at the survival of the patients in the graph below, the green line indicates that the survival of the patients with lots of T cells much, much better than the red line, which shows poor survival for the patients who do not have these T cells. So the question became, how can we drive the T cell responses into the tumor? What do we need to know about T cells in order to get these kinds of responses? And so T cell responses are actually very tightly regulated, and it's a complex process. And it took a while, many years actually, for us to really understand the process. So this cartoon diagram is simplistic, but I'll walk you through to show that initially the idea was it was the T cell receptor, which is this TCR, and that's signal 1. It's the T cell receptor interacting with the antigen in the context of the MHC molecule, and signal 1 should be enough to turn the T cells on. However, it was quickly realized that the T cell receptor by itself cannot turn the T cells on. And in order to have a truly activated T cells that can then generate a proper T cell response, you needed 2 signals. Signal 1 via the T cell receptor, and then the second signal was CD28 molecules, which then interact with B7 molecules on antigen-presenting cells. These 2 signals will then drive a T cell response. Once the T cells start the response, there are other genetic programs within the T cells that then become turned on. And these programs regulate another molecule called CTLA4. And eventually, CTLA4 is then expressed on the T cell surface, and CTLA4 interacts with the B7 molecules because it has a higher affinity and avidity for B7, which then replaces the CD28 signal. CTLA4 is now an inhibitory molecule that turns off the T cell response. So this is a cell-intrinsic process. The cell maintains homeostasis by having an on program, but also by having an off program. So the T cells are not allowed to continue to function forever. So Jim Allison and his group proposed that if the CTLA4 molecule is working in this inhibitory capacity, you can block this molecule and then allow the T cells to work for longer periods of time. So here you can see that the tumor cells are dying, and tumor cells can die for many reasons. They can die if you cut off the blood supply, as you heard in Dr. Jane's talk. They can die because of chemotherapy. They can die because of radiation therapy. And as they're dying, they're taken up by the antigen-presenting cell and presented then to T cells. But as you can see, the CTLA4 molecule will stop this response, and the T cells won't be allowed to completely kill off all of the tumor cells. By giving an antibody to block CTLA4, you can now have the T cells to continue its response and an enhanced anti-tumor response and tumor rejection. And Jim Allison's group showed this in many, many elegant mouse models that this concept worked. And not only did it worked beautifully in many, many different tumor types because you're not targeting the tumor cells. You're targeting your immune system. You're targeting your own T cells. And because of that, it can work for, the anti-CTLA4 therapy can work for many tumor types. The other thing was that he showed in mouse models was that the antibody can be combined with many, many other therapies. So you can combine it, for example, with the targeted therapies or with the therapies that target the vasculature that you heard about from Dr. Jane, or with chemotherapy and radiation therapy. So these studies were then taken to clinical trials in human patients. And initially in the phase 1 clinical trial, you could see this patient with metastatic melanoma. The red circles are these metastatic lesions in the lung of the patient. This patient also had subcutaneous nodules and brain metastasis, all of which went away after the patient was treated with anti-CTLA4 therapy. So it works in not only melanoma patient, it works in other tumor types. And this is, again, because you're targeting the T cells and not the tumor. And I'm not going to show you all of the data for the other clinical trials. But the point was we now had a better understanding of how T cells are regulated. And CTLA4 was only the first molecule. So CTLA4 led to a new understanding that we had to not only try to figure out what turns T cells on, but now we have to try and figure out what turns T cells off. And so the next pathway that was identified was the PD-1 and PD-L1 pathway, and this was then taken to clinical development as well. So I showed you the anti-CTLA4 responses in patients, and then in this next slide, we also used anti-PD-1 in patients, and in this patient was metastatic kidney cancer. And the top panel shows that as the anti-PD-1 therapy was given, this patient had a soft tissue nodule that then decreased in size and went away completely. The patient also had bone metastasis that completely went away and resolved after anti-PD-1 therapy. a biopsy was also taken of the lesion as this patient was on therapy. And initially, on the left panel in blue, there are very few T cells. However, after treatment with anti-PD-1, there were more and more T cells infiltrating into the lesion, as you can see the red staining. So we knew now that both anti-CTLA-4 and anti-PD-1 therapy could work, and they work through different pathways. So the thinking was that they can be combined, and Jim Allison's group did the combination therapy in mouse models, and then it was clinical trial was conducted and published by Jed Wolchuk in 2013. So here you can see again in melanoma patients, when they received both anti-CTLA-4 and anti-PD-1, the tumors shrank very quickly. In the top panel, you're seeing the time that it took for the tumors to shrink. Some tumors did grow, but a lot of the patients' tumors shrank. And the bottom panel shows the number of patients who had disease that was being decreased in size as a result of this combination therapy. So now we have very effective immunotherapy strategies, either as monotherapy or combination therapies. But the questions that arise then are, what are the cellular or molecular mechanisms involved in these anti-tumor effects? How can we figure out why some patients are responding and other patients are not? Are there potential biomarkers that we can look at? How do we combine these agents with other agents? As I mentioned before, they can be combined with chemotherapy and radiation therapy. And then are there other targets that we need to identify in order to have more and more patients respond to these therapies? And so to do that, we set up the immunotherapy platform at MD Anderson Cancer Center. And here we design the clinical trials so that we can obtain all of the matched tumor tissues and blood samples from the patients for the appropriate studies in the laboratory. And the clinical trials that are shown on the next slide. And basically, you can see that we had to rethink our clinical trial design. Initially, we all thought of phase 1, phase 2, and phase 3 clinical trials, which are the safety and dose escalation trial, the efficacy trials, and then the comparison to standard of care. However, we proposed these pre-surgical trials whereby the clinical endpoint of these trials truly are to get the tissues and the samples so that we can do appropriate laboratory studies to gain mechanistic insights and understand the biomarkers. The first clinical trial that we did this way was in patients with localized bladder cancer. And again, this was a trial, a very small trial, 12 patients. And the patients were all scheduled for surgery to remove their bladder. And we offered the patients a chance to participate in the clinical trial whereby they can provide informed consent to receive 2 doses of the antibody prior to going to surgery. And then we could collect all of the tumor tissues at the time of surgery to understand the impact of anti-CTLA-4 in the tumor microenvironment. We also had another cohort of patients who did not receive the antibody, who did have surgery, and that was the control cohort of patients. There's another clinical trial I'm showing here, which is combination therapy, because we're interested in how to combine these immunotherapy agents with standard of care therapies. And in prostate cancer, the standard of care therapy is hormonal therapy, such as Lupron. And here we combine anti-CTLA-4 with hormonal therapy in 20 patients that had localized prostate cancer. These clinical trials then provided access to all of the tumor tissues so that we can do in-depth immune monitoring in both the matched tumor samples and the blood samples, as shown in the cartoon here. So initially, we looked at the immunohistochemical studies from these tumor tissues, and what we can see is that in the patients who had no treatment compared to patients who received anti-CTLA-4, and in this setting I'm showing the prostate tumor tissues, we saw the same thing in the bladder tumor tissues, that there were a great deal of significant increase in the T cells. So here there are CD4 T cells, CD8 T cells, and granzyme B, which is activated, shows activated T cells. These are all increased after therapy as compared to no treatment in the control cohort. The other analyses that we can do is a gene expression array looking at the untreated tissues and the treated tissues. And since I just showed you that the treated tumor tissues had a lot more T cells, the gene expression data actually showed a lot of T cell pathways. And so at the top of this pathway, the top pathway identified was called the ICOS pathway. And so I'm going to speak a little bit more about that. But all 10 of these pathways actually were related to T cells being within the tumor tissues. So the ICOS pathway is actually another family member of CD28 and CTLA-4. And what we did was what looked at the tumor tissues by flow cytometry to confirm the gene expression data. So here by flow cytometry, the non-malignant tissues in the bladder cancer setting, as well as the untreated tumor tissues and the anti-CTLA-4 treated tumor tissues, and then in the prostate cancer setting, the untreated tumor tissues and then the anti-CTLA-4 plus hormonal therapy treated tumor tissues. We looked at the ICOS subset in CD4 T cells, and in the non-malignant tissues or untreated tumor tissues, they're very few. They're about 13 to 16% here actually, and 14% in the prostate cancer setting. However, after treatment with anti-CTLA-4, there's this increase in frequency of ICOS-positive T cells. And so ICOS was truly increased within the tumor tissues as a marker for anti-CTLA-4 therapy. And we asked the question now, are these ICOS-positive T cells associated with clinical benefit? And to do that, we did a retrospective study in a cohort of melanoma patients who had received anti-CTLA-4. And these patients, we found that if the patient had sustained increase in ICOS-positive T cells, they had much better survival, that's the red line, compared to the patients in the black line who did not have an increase in the ICOS-positive T cell subset. So this led us to a hypothesis that the ICOS ligand pathway is really playing a role in the anti-tumor response, and that this pathway can be targeted to improve the anti-tumor responses in the setting of anti-CTLA-4 therapy. And so we did this study targeting ICOS pathway in combination with anti-CTLA-4 in mouse models. And here you can see in wild-type mice, which is a WT, monotherapy, if you treat with just anti-CTLA-4 alone or an agent to target the ICOS pathway, monotherapy gives you about 20% of these mice have rejection of their tumors and long-term survival. However, if you give combination therapy, now you can see that about 90% of the mice have tumor rejection and long-term survival. The same experiments that we did in wild-type mice were conducted in the ICOS knockout mice, and these mice lack the ICOS gene. And here you can see that you lose the ability then to have this improved anti-tumor response. So we identified ICOS as both a pharmacodynamic biomarker and as a new target for combination therapy. And it's very important to think of combination therapy because I think for many years cancer patients were dealing with the idea that we had standard therapies such as chemotherapy that can allow for some improvement in the median survival, as you can see between the black and the blue line. But we were not really getting control of disease and long-term responses because all of the patients still died. But with immunotherapy, we can now see that you have about 20% or so of patients. We've seen this with the anti-CTLA-4 studies. With patients for 10 years, about 20% will live 10 years or more. So they're practically cured, these patients, as a result of immunotherapy. And so you're getting long-term and durable survival, which we can then improve, which is the red line, with combination therapy. So our goal should really be to now get more and more patients to have an anti-tumor response by including immunotherapy as the backbone of our treatment for cancer patients. And that's definitely becoming the new way to move forward as more and more of these targets are being identified. So this cartoon is just showing all of the different immunotherapy targets that are now identified and being developed clinically. So I would just like to conclude now and say that pre-surgical clinical trials truly are a feasible platform where we can study the tumor tissues and understand the biologic impact of different agents, and in this setting with immunotherapy agents. And CTLA-4 blockade leads to measurable immunologic changes that we can see now as a pharmacodynamic biomarker and as a new target in the form of ICOS. And immunotherapy does provide a rationally designed treatment modality to induce long-term clinical benefit and survival for decades. And it's definitely joined the ranks of surgery, radiation, and chemotherapy as a pillar of cancer treatment. So on that note, I'd like to thank the many people actually who did the work, because the types of study I just showed are never done by a single person. So my lab is very much involved in doing the animal studies, and of course the clinical trials we conduct with our clinical team in GU Medical Oncology. And of course I really do want to thank the patients, because these patients participate in the clinical trials to allow us then to have access to the tumor tissues and the blood samples for the analysis. So on that note, I'll end and say thank you, and I'll take questions.
Okay. Well, thank you, Dr. Sharma, and also thanks to Dr. Jain for your very engaging presentations. We're going to move along quickly to some of the questions that have been submitted by our online viewers. As a quick reminder to viewers, You can still submit your questions by clicking the Ask a Question button below a slideshow, type your question into the box, and then click OK. So since we just finished up your talk, I'll address my first question to you, Dr. Sharma. One of the viewers asks about FDA-approved anti-CTLA-4 drugs. Are there any that are FDA-approved, or are these still experimental?
No, so there are FDA-approved. It's called ipilimumab, and so that's a Bristol-Myers Squibb drug. It is FDA-approved for treatment of patients with metastatic melanoma. Anti-PD-1, nivolumab, and that's from Bristol-Myers Squibb as well, and anti-PD-1 pembrolizumab from Merck are also FDA-approved drugs for patients with metastatic melanoma. And there are clinical trials ongoing in non-small cell lung cancer, renal cell carcinoma, bladder cancer, prostate cancer. So we expect that these drugs will also have efficacy in these other tumor types, and look for FDA approval hopefully in the next few years for these cancer types as well.
And there's another question about the timing of the therapy with the immune checkpoint inhibitor. How long do you have to administer that sort of treatment? So
anti-CTLA-4 right now is FDA approved so that it's given as 4 doses. So you get dose number 1, 3 weeks later dose number 2, 3 weeks later dose number 3, 3 weeks later dose number 4, and then you're finished. So that's it. 4 doses given 3 weeks apart.
Do these immunotherapy approaches increase the likelihood of developing autoimmune disorders or something like that?
You definitely see immune-related adverse events, and most of them can be treated by stopping the drug and by giving corticosteroids. So we don't really call them autoimmunity, because autoimmunity is sort of this chronic, ongoing problem that, if you stop the drug and give corticosteroids, the patient's adverse event does go away, the toxicity does go away. Not for all of them, but the majority of them. So the few that do not are hypophysitis, but things like diarrhea, colitis, hepatitis, pancreatitis, those do resolve. So they're not true autoimmune events.
Okay. So I think I'll move on to asking some questions to Dr. Jain now. So we've had a lot of questions from our online viewers, but of course we only have time to ask a few. So I'll start off with this first one that's kind of a general question. Is there any risk of enhancing tumor growth by normalizing a tumor's blood supply?
Yeah, there is a risk if you're not giving any cytotoxic therapy with it. So when you normalize blood vessels, the goal is not only to alleviate hypoxia, but also if you were to give a chemotherapy with it, or radiation therapy with it, or immune therapy with it, the delivery of these drugs, these therapies would improve, and it will kill more cells. Does that answer the question?
I think
so there will be 2 benefits for normalization. You'll improve the delivery, and the second advantage will be you'll alleviate hypoxia. So the drugs that get in there, whether they're chemotherapeutic drugs or immunotherapeutic agents or radiation, they will work better.
Right, right. Just normalizing the blood supply alone is not helpful.
Well, I don't think so. Unless the immune system is so profound that the added immune cells of the host, they get in and do the job, but I would not take that chance, or at least until we understand more about immune therapy.
Right. So I have another question here about tissue specificity. Do the effects of normalizing blood flow in enhancing cytotoxic therapies differ between different tissue types, depending on where the tumor is located? Are different tissues differentially responsive to that approach?
Yes, different tissues do respond differentially, and the reason for that is that the levels of VEGF in different tissues may be different. So if you give a certain dose of anti-VEGF agent, that may normalize vessels in, say, organ 1 metastasis, while in organ 2 metastasis, it may not be enough or it may be too much. So you need to look for a sweet spot. And that's why a lot of the trials we did, we looked at this with imaging. And we are looking for systemic biomarkers right now for normalization. And we have some candidates, but we don't have anything validated.
I have a question here about the drugs that can be used. Are there any drugs that act on the renin-angiotensin system besides losartan? Anything that you've tested that can have the same effect on improving blood flow to the tumor?
Yeah, so there are 15 drugs in that general class approved by FDA. The reason we chose was losartan was because for multiple reasons. One is safe. It's been given to millions and millions of patients worldwide. It's very inexpensive, about $100 per year, approximately. And its safety profile is pretty good. And it has been tried in Marfan syndrome. There's a clinical trial going on from Johns Hopkins on that. So that also suggests that this may be worth doing. But currently, we are looking at other ARBs, other angiotensin receptor blockers. And as I mentioned in my disclosure, that some of this work we have done in this area has been licensed to a company, Extuit, and I am a co-founder of it, and I own equity in it. So this is just for full disclosure. I want you to know that. So we are developing drugs which hopefully are much better than approved blockers of angiotensin pathway. Because one of the big challenges with these blockers is that you need to deplete stroma. You need higher doses, and if you give higher doses, you're going to cause lower blood pressure, and you don't want to do that in these patients. So we are developing drugs which do not lower blood pressure, because in 2011 we published a paper showing that the amount of depletion of the stroma is very dose dependent, and then when we increased the dose to a mouse, we found out there was a decrease in blood pressure, which was not acceptable. So that has led to this whole new set of work. And there was also a trial published from Japan a year ago, where the patient received candesartan, another antihypertensive drug. And what that trial showed was the patients who could tolerate or had a high dose of these drugs, they seemed to have better benefit than the ones that could not be given higher doses. So that's the direction our current research is heading.
And then I think I have one final question here that can kind of be addressed to both of you, I think. How much or does the reduced blood flow to a tumor affect the ability of things like the anti-CTLA-4 antibodies to get into the tumor microenvironment, or is it just sufficient that the anti-CTLA-4 is in the body? Because
I will let Dr. Sharma answer it, and then I will
receive the signal, right?
Yeah.
I'm wondering about how much the impeded blood flow in a tumor impedes the ability of the T cells to get in there.
So we haven't looked at this directly to say whether or not it's a change in blood flow. When we give the anti-CTLA-4 therapy, both in mice and in humans, you can definitely see difference in T-cell infiltration. So, and obviously the T-cell infiltration is occurring as a result of blood flow, but whether or not certain tumor types may have impaired blood flow, I should say, then that's why the immunotherapy may not be working as well in certain tumor types. We don't have all those answers yet. But currently in the tumor types where they're working, we haven't really looked at blood flow as the issue. We do see a change in the T-cell infiltration, and that seems to correlate with the anti-tumor response and tumor rejection for both the mouse models and the humans.
So we have done mouse studies, and where we published this in, I think, a couple of years ago, where we lowered the perfusion. Actually, I showed that slide when we lowered the dose of anti-VEGF agent to 1/4 of the standard dose to 10 milligrams. In those animals, the tumor perfusion went up, and the delivery of CD8 cells also went up. And those animals had a better response to immune therapy. So at least in the murine setting, we are seeing it clearly. But remember, blood flow is not the only thing. If the tumor is highly desmoplastic, even the blood vessels are open. If there's a highly dense matrix, such as in pancreatic ductal adenocarcinoma, for example, then these T cells or the immune cells have trouble penetrating these tumors. And there are studies done by others and us where we have shown that if you do the matrix normalization, you can improve the delivery of both anti-CTLA-4 and anti-PD-1 antibody, and we see a better response and better survival. Again, murine studies. We would like to test this in patients, and we are discussing this with our collaborators at various Harvard teaching hospitals to test this hypothesis in patients.
Well, I think that is going to about do it for time. I think we're out of time now. I'd like to sincerely thank Dr. Jain from Harvard Medical School and Dr. Sharma from MD Anderson Cancer Center for being with us today and for their fantastic talks and the discussion. Many thanks to our online audience for the questions you submitted to the panel. I'm sorry that we didn't get time to get through more of them. We got a lot of those. Please go to the URL that's now shown at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation within approximately 48 hours from now. We'd love to hear what you thought of the webinar. You can send us an email to the address that's now shown in your slide viewer, webinar@aaas.org. Again, thank you to our panelists and to Cell Signaling Technology and the Koch Institute for their kind sponsorship of today's seminar. Goodbye.